So this is my video on mitosis versus meiosis. First, we're going to start off with mitosis. Mitosis is for identical cell replication. That is really important because you're trying to make an exact copy of the same cell that obviously it was there before. Your nucleus is what is going to hold all of your DNA. Your DNA is your genetic information. And you have 46 chromosomes at first in each cell. So you'll have 46 chromosomes. However, after you go through interphase, which is where your DNA replicates, the cell grows, or it goes through cell function, and you'll see growth. That is where you will see your 46 turn into 92. So here's one, and you'll see the addition to make the X pair for your 46 chromosomes. So first phase is interphase. Then you're going to go through PMAT, which prophase, metaphase, anaphase, and telophase. So for mitosis, you only go through your different steps one time. For meiosis, however, you're going to go through these steps twice. So you'll have to label prophase 1, metaphase 1, anaphase 1, telophase 1. And then for the second round, prophase 2, metaphase 2, anaphase 2, telophase 2. So for prophase 1, this is where you'll have the condensing of chromosomes. So these guys are inside of a nucleus that is starting to kind of dissipate. And then you'll have 46 of these paired guys here. And they're all kind of free floating at this point. During metaphase, however, the chromosomes are going to line up in the center of the cell. So the nucleus is going to dissipate, and you'll have 1, 2, 3, and 4 that are lined up in the center. During anaphase is where the chromatids are going to separate with the spindle fibers that are starting to form here. So during anaphase, you will see that these spindle fibers are going to attach to each side of the chromosome and split them apart, the chromatids. And then split by spindle fibers. And then during telophase is where you will see that the chromosomes are going to be in a newly formed nucleus. So these guys are over here, 1, 2, 3, 4. These guys are over here, 1, 2, 3, 4. And then cytokinesis is going to happen, and the cell wall is going to pinch off to create 2 separate cells with identical genetic information for each side. This is where you will find your 23 and 23 with 46, or I'm sorry, not 23, with 46 on each side. So you get 46 because back here in interphase, you had DNA replication, which took your one cell, turned this 46 into 92. So we have 92, 92. You see the split off to 46 and 46. For meiosis, however, you will see that in the beginning, you will go through interphase again. So that's where the DNA replication will happen. You start off with meiosis. You have gametes, which is your egg and your sperm. Each of these originally has 46 each. So we'll say this is the egg and the sperm. Both are gametes. So this egg cell here, you need to eventually split into 4 different sex cells, each with 23 chromosomes, so that they can pair with the opposite sex cells' 23 chromosomes to form together to make one 46 chromosome person. So, your egg, you have 46. You go through interphase, which is DNA replication. So your 46, again, gets turned into 92. Same thing that it was with mitosis. Same thing here for meiosis. So, after you have your 46 chromosomes and 92 chromatids, you're going to go through prophase 1, metaphase 1, anaphase 1, and telophase 1. After you go through all of these steps, same thing as it was with mitosis, with meiosis, then you are going to have prophase 1, where you have your chromosomes floating around all over the place. Metaphase 1, they line up in the center. And then remember, with meiosis, you're going to have twice as many inside of this original cell. So here, you're going to have 1, 2, 3, 4 on this side. 1, 2, 3, 4 here. And then what's important with meiosis is that you have homologous pairs of chromosomes that are going to match up in the center of the cell, and they're going to interchange genetic information. This is how you get a difference in the ending 4 sex cells, so that you could have 2 babies with different genetic information come out of the same mother and father. It won't always be the same coming from the sperm and the egg. Otherwise, we would all be identical to our siblings. So, for prophase 1, everything matches up, and you see the homologous pairs interchange genetic information. During metaphase 1, the chromosomes are going to all line up in the middle of the cell with the pairs. And anaphase 1, the chromosomes are going to be pulled away by the spindle fibers. Notice I said chromosomes, so these pairs are going to split apart. So the spindle fiber is going to each have 1 X instead of just 1/2, like it was in mitosis. So once you have gone through telophase 1, this imaginary line here, you're going to go through cytokinesis. Which is going to form your 2 nuclei. inside of the cell. Cell wall is going to pinch off to create 2 with your 46 chromosomes in each nucleus. Now you have 2 coming from your original. These 2 now are going to go through this same process again. So it's going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2. So, during prophase 2, your spindle fibers are going to start to form. In anaphase 2, in metaphase 2, the chromosomes are going to line up. So we have 1, 2, 3, and 4. This is the step that's a lot different from metaphase 1 because you don't have the pairs. You just have one set. The spindle fibers are then going to attach and pull apart the chromosomes into the chromatids. So during anaphase 2, now you're going to have your spindle fibers in your cell, each having 1/2 of your pairs. In telophase 2 is where you're going to see the nuclei form. And these are going to be inside here and inside here. Imaginary wall. Cytokinesis is going to happen, which is going to pinch these off into 1, 2, 3, and 4 cells. Because remember, this one here came down. This one here is also going to do the same thing. So this is going to be times 2. This is going to be times 2. This is going to be times 2. And thus this times 2. So your end result is going to be 4 sex cells, each with 23 chromosomes. Because here we have 92. 92. We split the 92 into 46. 46. 46. Here we see the splitting, pulling apart to 23 and 23, 23, 23.